We can even show the molecules that were imaged to make that call. And all of that data can also be exported as a standard PCF file, which allows you to use any software that you may have. Our software is a built-in control database of hundreds of normal controls. And so when you get the ST calls for a genome like the ones you see here, there's about 5,000 structural variant calls. We see them as these mostly orange and green dots in the outermost circle of the circus plot, and those are insertions and deletions, and then the more rare inversions and translocations, duplications are shown in the middle. The copy number tool is shown, as well as the innermost bands there. Now, with a single click, you can remove variants that are seen in this large control database. And so from 5,000 structural variants or so, you just go to the 50 or so that are specific for your sample, as you see here, far fewer dots, and that makes analysis a lot easier. And our software will show you which genes they overlap with, whether they've been seen in the control database or not, and at what frequency, whether they're homozygous or heterozygous, etc. And so compared to sequencing technologies, Bionano has really the unique advantage of having extremely long molecules. The average is about 300 kb, but we go as long as 2.5 megabases, and that's really orders of magnitude more than even long-read sequencing. And that allows us to call these structural variants with much higher sensitivity, but also at a very high throughput. We can run 12 human genomes per day per instrument, or if you run cancer samples 3 a day at high coverage of 400x. And as I mentioned, we can go as high as 1600x. That allows us to find structural variants, even in heterogeneous cancer samples at just 1% allele fraction. All that for just a few $100 per sample. Long-read sequencing technologies just cannot match that output and throughput. Now, if you want to know more, if you want to see other examples, please look at our website, bionanogenomics.com, and have a look at the video section, because we have webinars such as this one and hundreds more recorded on just about any genetics topic you can think of. So with that, I want to turn it back over to Frances, who's going to tell us about her study on CDH.
Thank you, Sven. So I'm going to talk about our study design. We chose samples from a mix of patients with known or suspected structural variations. And so these included patients with deletions, duplications, and translocations, as well as what we call unknowns, for whom a causative genetic variant had not yet been identified. There were several goals of this study. The first goal was to confirm known structural variants and to gain additional insights into a higher order structure to help refine pathogenesis. The second goal was to define new CDH candidate genes in genetic loci through the identification of novel CDH-associated structural variants. Another third goal is to compare structural variant calls between different technologies, including array CGH and whole genome sequencing. So we actually did this in 2 different batches. We had a preliminary cohort, which was just 19 patients, and that included 5 trios. These are patients along with their unaffected parents. I'm going to talk to you a little bit about the power of the trio approach. So that allows us to identify de novo structural variants, or de novo structural variants are thought to be more likely to be pathogenic versus those that are inherited. We also had 12 singletons and 2 patients from multiplex families. These are families that had more than one affected individual. In this first cohort, 10 of the patients had previously identified high confidence structural variations that had been detected by standard clinically kind of standard of care technology, including array CGH and karyotype. But we also included a small number of patients that were unknown in this preliminary cohort. The follow-up cohort, which we are still in the process of analyzing, is 50 patients. They're all trios, so we did both 50 patients and their parents as well. They're all unknowns, and they also have all undergone whole genome sequencing, and some of them have undergone array CGH as well. So I'll just summarize the results. From our preliminary cohort, this analysis is complete. That's the 19 patients. We confirmed all high confidence structural variants that were detected by array CGH or karyotype. And I'm going to show you some examples of cases where we saw an additional level of detail for some structural variants. that using the Bionano technology provided this additional level of detail over array CGH or karyotype. And we also identified at least one novel likely causative structural variant in a patient that had not yet been studied by array CGH. As I mentioned, we're in the process of analyzing the data from our follow-up cohort, but I do have some interim results, and I'll share a few examples of cases with you today. We've completed the analysis on 37 of the planned 50 trios, and we've done an analysis focused on looking at de novo structural variants, rare inherited structural variants, those that have a frequency of less than 5% in that control database, and we focused on variants that have overlap with known or candidate CDH genes in genomic risk regions. So I'm just going to jump into talking about some of the cases to highlight nicely some of the ways in which you could use the Bionano technology to study specific types of variants. So I have a total of 7 cases to go through. This is the first case. This is a case of a patient who had a known de novo balanced translocation. So de novo, this was a balanced translocation that was present in the proband. It was not present in either of the parents. And when we see these de novo balanced translocations, we think to ourselves of the possibility that the causative genetic factor may be at the break point between the balanced translocation. Even though there's no missing genetic material here, sometimes that break point can disrupt a region. And that was our hypothesis for this patient. So using the Bionano technology, we were able to map the 2 break points for this chromosome 1/6 translocation and identify some potential putative candidate genes. So we looked at the breakpoint on the derivative chromosome 1, and it was in a gene core region. It wasn't really worth pursuing. But the chromosome breakpoint on chromosome 6 was quite interesting, and it was breakpoint was just upstream of this gene VGLL2. And when we look in the ENCODE database, we can see that this region actually contains a number of putative regulatory regions. We were interested because VGLL2 is an intriguing gene. It functions as a transcriptional co-activator in myogenic differentiation, and it's strongly expressed in skeletal muscle. Of course, the diaphragm is a skeletal muscle. So we hypothesized that this gene may be playing a role in CDH pathogenesis, and we're in the process of investigating this further in some functional experiments in our laboratory. The second case that I want to talk about is a case in which we used Bionano technology to try and understand a complex structural rearrangement in a part of the genome that's challenging to study. And so we identified that duplications at this locus, 16p11.2, are a CDH risk allele. And this was published back in 2018. And at that time, we had used microarray technology to study a large cohort of CDH patients and compared them to population controls. And we found duplications at this locus that were enriched with statistical significance compared to controls. So we wanted to try and understand what was going on at this locus in more detail. So we chose one of these patients to study with Bionano. And the result was quite intriguing. So the array CGH or microarray technology showed that there was one large 2.3 megabase duplication at this region, but Bionano actually resolved that this variant was considerably more complex than that. So what you can see here is the reference genome in green and the patient genome in blue. And at the top, in the sort of lavender color, is the copy number call. And what you can see is that this is actually 2 adjacent duplications instead of one large duplication. And that the second duplication is actually inverted as well as duplicated. So this rearrangement ended up being considerably more complex than we had originally hypothesized based on the microarray technology. So this is just another depiction of this rearrangement, and we were able to identify the genes in this area. So this helped us refine which of the candidate genes in this area that we may consider being involved in the pathogenesis of CDH. It's important to note that this is a region of the genome which involves extensive segmental duplications, which is one of the reasons why it's been challenging to study with other technologies, and one of the reasons why Bionano has been helpful for this particular rearrangement. In the next example, I'm going to tell you about, this is a multiplex family, so you can see a pedigree here. And this family was studied by microarray. The individuals in black are affected individuals with CDH and also agenesis of the corpus callosum. And individuals in this family were shown to have what looked like a duplication at chromosome XQ13.1. This region includes really one candidate gene of interest, which is EFNV1, otherwise known as FNV1. This is an interesting gene. Mutations in this gene cause craniofrontal nasal dysplasia, which is an excellent dominant condition that is rarely associated with CDH. About 10% of patients with mutations, so loss of function mutations in this gene, have CDH. But there are this family, as well as two other families that have been described, show copy number gains of this region that appear to cause a distinct syndrome, different from craniofrontal nasal dysplasia, and that these families have a high penetrance of CDH. So we wanted to study this rearrangement in more detail. So what was quite interesting is when we used Bionano to study an individual from this family, what we see, if we look at the copy number profile that's in the top in lavender, is that there's a copy number gain here, but it appears to not only be a duplication, but actually a triplication. So you can see a copy number of 3.8 on average in that call. And if we detect on, on a more molecular detail, what we noticed was that this is not only a tandem triplication, but that one of the repeats inside this triplication is inverted. And we were able to, this is really something that we really needed the Bionano technology in order to identify at this level of structural detail. This is case number 4. This was a case from our new cohort that shows an interesting de novo translocation between chromosome 11 and 13. So this is a circuit plot, as Sven said, that this is one way in which we can look or visualize the variants using the Bionano software. And what you can see, this line, this pink line going from one side to the other, shows the translocation between chromosome 11 and chromosome 13. This translocation is interesting. So there are actually, it's associated with a copy number gain on both chromosome 11 and chromosome 13. And so this slide just shows the mapping of the breakpoints at both chromosome 11 and chromosome 13, so 11 on the top and chromosome 13 on the bottom. So we can identify the location of the breakpoint, and we can also identify in this lavender color the copy number gain on chromosome 11 and chromosome 13. And I'll mention also, it was shown in the previous slide, but there are actually some interesting CDH candidate genes that are in both of these regions involved in the duplication. I'm now going to sort of tell you a few examples of cases that involve a region that we know is involved in CDH pathogenesis, and that is a region on chromosome 15q. So I'm going to show you two cases of translocations that we were able to study by Bionano involving this region, and then also a case involving another structural rearrangement that helped us refine the pathogenesis of the candidate genes within this region. So this is case 5. This is an unbalanced translocation between chromosome 1 and chromosome 15. You can see this the circus plot here, and that pink line again going across that shows the translocation between chromosome 1 and chromosome 15. This results in a derivative chromosome 15 with a deletion at the Q terminus and a corresponding duplication of 1Q. This is the second translocation that we identified. So in this case, this was a trio. And so I'm showing you 3 circus plots. The bottom is the proband, and then the top are the father and the mother. And what you can see is there's a translocation between chromosome 11 and 15 in the proband, which is also present in the father. If we look at these individuals in more detail, if we look at the father, we can see that this is a balanced reciprocal translocation between chromosomes 11 and 15. And so these are the breakpoints here on chromosome 11 and chromosome 15. But you can look at, if you look at the copy number at the top, you can see that there is no copy number gain or loss on either of these chromosomes. This makes sense. The father is healthy. He doesn't have CDH. However, in contrast, the child carries an unbalanced version of this translocation. And you can see here, if we're looking at this is again the breakpoint on chromosome 15. And what you can see is that this downstream region, the child shows a significant deletion of this portion of chromosome 15. So this region is quite interesting to us in chromosome 15q26. So back in the early 2000s, this gene was first postulated to be a recurrent chromosome hotspot for CDH, and there have been a number of papers that have been published identifying patients with deletions in this region. And people have speculated on which candidate regions in this recurrent deletion region may be responsible for the CDH phenotype. And there are a number of different genes that have been proposed. I've just mentioned the ones that are most commonly noted here. So I'm going to focus in on this gene, NR2F2. So this was another interesting case that we identified from our cohort that we studied with Bionano. So this is a patient with CDH that has a complex structural rearrangement of chromosome 15q26 that involves the NR2F2 gene. So in contrast to the other cases that I just showed you where there is a large deletion including multiple genes, this is instead a structural rearrangement that seems to be specific just to the NR2F2 gene. And so to just walk you through this a little bit, we've got the reference genome in green, and then the maps from the patient in blue. And so on the bottom blue map, we can see that there is a large deletion of this region that includes the NR2F2 gene. And then there's also an inversion of this region just upstream of the, or downstream of the NR2F2 gene. And then there's another smaller deletion just beyond that. So just to put this in a different graphical form, if we focus in again on chromosome 15q26 at the NR2F2 locus, this is a wild type or typical allele. We have NR2F2 as well as an antisense gene which overlaps with it. And in our patient, we have a deletion of this region, including both NR2F2 and the antisense gene, and then an inversion of this downstream region detected by the triangle with the number 1 on it. So this was a very interesting structural variant to us because we're interested in NR2F2 as a gene from our sequence studies. So NR2F2, which was previously known as COOP-TF2, is a transcription factor that acts as a co-repressor of the retinoic acid signaling pathway. The retinoic acid signaling pathway has been shown in a number of different studies to be one of the core signaling pathways involved in the pathogenesis of CDH. And so this was a very interesting candidate gene for us to be the major causative gene involved involved in 15q26 deletions as a causative factor for CDH. And we and others have identified de novo sequence variants in cases with human CDH. And so now by being able to study structural variation and showing a structural variation that also involves the same gene, it really adds an additional level of support for this being a true pathogenic CDH risk factor. This gene is also linked with congenital heart defects, and there is evidence from a mouse model that shows that this role for this gene in CDH. Those were the major examples that I wanted to show you. And I just want to finish by concluding that structural variants are important for the pathogenesis of congenital diaphragmatic hernia. That I hope that I've shown you that optical genome mapping provided an improved level of resolution of some structural variants compared to array CGH, and that these systematic analyses have identified and or validated recurrent pathogenic structural variants in CDH. And I've shared the example of 15q26 deletions. And that I hope that analysis of structural variation and sequence variation in parallel will result in identification of critical genes and pathways for CDH. And I think we're beginning to see this already in the data we've shown so far with the example of NR2F2. I would also like to point out that I think that these investigational strategies can be applied to other structural birth defects, not only structural birth defects, but other similar genetically heterogeneous conditions. And in the future, we're really hopeful that direct comparisons of these different platforms for assessing structural variation will be useful to determine which technology or combinations of technologies will be ideal for ongoing studies. And so we're, as I mentioned, in the process of analyzing our whole genome sequencing data from the same cohort. So stay tuned, and I think we're going to be able to see a direct comparison of these different technologies. And with that, I just want to thank a lot of people. I want to thank the members of my team at Mass General Hospital who help recruit these patients and do all the follow-up functional analyses in the lab. I also want to thank the team at Boston Children's Hospital, where we also recruit a large number of our patients. France Petit at the Hôpital Jean de Planta, who is my collaborator on the EFNG1 case that I showed. And I'd like to give a special shout out to the Bionano Genomics crew, particularly their analysts who've been really helpful in teaching me as a non-bioinformatician how to use this software and how to do these analyses. And it's really been, they've been really helpful. And then finally, my funding sources. And with that, I think I'm done, and we should have time for questions.
Yeah, thank you, Francis, for your insight into CDH and how advanced structural analysis of risk loci can improve our understanding of the disorder. And thank you to Sven as well. We've received a number of questions from audience members, so we'll try and get through as many as we can in the time remaining. But please be assured that any questions that aren't answered live today will be answered by email after the event. Okay, so our first question is, could you use DNA of a non-neoplastic tissue as a reference genome for somatic non-neoplastic changes, i.e. buccal mucosa relative to bone marrow?
So I think that's a really interesting point. So the question is really about somatic, trying to assess for somatic changes, and that's something we've been interested in CDH, because there's a possibility that maybe some of the the causative genetic changes in CDH are actually specific to the diaphragm, which is where the localized defect is. So I could imagine we certainly could apply this technology to a diaphragm specimen from one of our CDH patients. And sometimes we can actually get that tissue at the time of surgical repair and see if there are any structural variants that are seen in a somatic, in a somatic way, when comparing that to, for example, a blood sample from the same patient.
Okay, thanks. And in today's world, the size of biopsies are getting smaller and smaller, and several molecular methods are required to diagnosis. How much input DNA is required?
Maybe I can answer that question. So for solid tissue, we require 5 milligrams of material, and that can be fresh or flash frozen. What's important here, though, is that FFPE preserved samples are just too fragmented. The DNA is too broken from this cross-linking, and it's not suitable for this analysis. But for solid tissue, we require 5 milligrams. For blood samples or bone marrow, less than 650 microliters.
Okay, thanks, Ben. And the next question is, you seem to focus on structural variants in these genes. How much do they actually matter compared to single nucleotide variants?
That's a good question. So I think the bottom line is we need to study both in parallel, and that's the way we've designed our study. So I shared the statistic that in CDH, we know that at least 10 to 15% of cases are caused by a structural variant. And so keeping in mind that percentage, if we only did sequencing technology on all of our patients, we would miss all of those, all of those causative variants. So you have to both in parallel.
And you showed a number of interesting variant genes. Are you performing any functional studies to determine if these genes are causative to CDH?
Yes, absolutely. So we have a whole branch of the lab that's performing functional variants. We have a number of different assays up and running. We do a lot of mouse modeling. So a number of these genes are being modeled in mouse knockouts. And then we also have some cell-based technologies to try and model not only the development of the diaphragm, but also the development of the lung, which we know plays an equally important role in the pathogenesis of this disorder. I didn't share any of those with you guys today, but perhaps for another talk.
Okay, great. I'm afraid we're going to have to put it there. Again, let me reassure you that any questions left unanswered today will be answered after the event by email. This webinar will also be available on demand in just a few days' time, and you'll receive an email alert when it's ready. Thank you once again to Francis and to Ben as well for presenting, and thanks to everyone for joining us today. I look forward to seeing you. Actually, we've just had another question come through. If you don't mind answering that one, Francis. Are there any differences on how tissue is collected and or prepped for use with Sapphire versus the other technologies you discussed? Apologies for the last minute one there.
Yeah, that's no problem. So I think Sven talked about this a little bit, but the source of DNA for Sapphire has to be slightly different because you need these long molecular weight DNA molecules. So he talked a little bit about we need a fresh blood sample or a fresh tissue sample. So that's the main difference. So in our particular case, we used cell lines, just because we had generated them through our study. But you can also use fresh blood, and it just can't be the same extracted DNA that you would typically use for genome sequencing.
Okay, great. And how many chorion tissue or amniotic fluid is necessary for prenatal analyses?
Maybe I can address that. So right now we need to work with 1.5 million cells total. So that means that these still need to be cultured. We are working on reducing the input material, but right now that is the requirement. So 1.5 million cells.
Okay, great. And do you see optical genome mapping as a first-line test for your patients, or do you still, or do you think you need to ACGH still?
So I personally think that this could replace ACGH and karyotype. And I think that the data that I showed shows that we were basically able to identify all of the variants that were detected by our ACGH and by karyotype in using the Bionano Sapphire platform.
Great. And is it possible to completely rebuild an individual genome with Sapphire data?
Ben, do you want to comment on that?
Sure, yeah. So we didn't really go into much detail about that part of the process, but when these molecule images are collected, our software will perform an entire de novo assembly of the genome, meaning that every molecule that was imaged is aligned with every molecule that was imaged, and the software will build these overlap graphs that span entire chromosome arms. So we will build, or our software will build, the entire genome structure completely de novo on every single sample that is analyzed. And then once that genome is built, it can be compared against a reference to call structural variant. So yes, the whole genome gets built.
Okay, great. Again, sorry, we are going to have to leave it there. Again, as I said, any unanswered questions will be answered by email after the webinar, and it will be on demand in a few days. As I said, thank you once again to Francis and to Sven for presenting, and thank you to you all for joining us today and for your questions. I look forward to seeing you at future webinars. Goodbye.
Okay. Oh my goodness. Yeah, I'm surprised with the share price today, man. Oh man. If we do manage to get this though, oh baby. Imagine. Just imagine, guys. I need to start working on another video for tonight. I'm piecing together some stuff on a summary of yesterday and some prenatal stuff. I've got some throat burps. But yeah, guys, thank you very much for tuning in. Sorry I got it up so late. I was in a lecture. I was in an online lecture literally minutes before, so I tried to set up the stream quickly. But yeah, thank you very much, guys, for tuning in. Ooh, I got some throat burps. But yeah, I'll catch you guys in a bit, and hopefully you'll see the video tonight. I hope you enjoy it as well. But I'm long and strong, baby. I'm long and strong. I'm going to actually be averaging down. I'm hoping it comes down a bit more, because I'm surprised there's that kind of sell-off reaction. But it seems like the whole market, I was looking across genomics as well at Pack B and Invitae, and they're both down as well today. So Invitae is down 5%. Pack B is down 4.2%. So I'm not surprised. But yeah, thank you very much for tuning in, everybody, and I'll catch you next time. See you later, Ash. Mr. Invest a Lot, over and out, baby.